Hey, hello everyone. We will wait a few more minutes to other participants to join, and then we will start. So in 5 minutes we will start. Okay. Hello everyone, one more time, welcome on our webinar about AQUA-DUCT. The webinar is instead of summer school. We wanted to prepare it for this summer, but due to pandemic it was not possible, so that's why webinar came. And before we'll start with description of the software and with what you can do, you will let me introduce our group. So we are
we are having some technical problems.
Small problems. Just 1 second. Yeah, just 1 second. Just 1 second. Okay, we are. Okay, so let's start from, my name is Artur Góra. I am head of the tunneling group, and we are located in Poland, in the middle of the Europe, and in Silesian University of Technology and Biotechnology Center. Our group is quite new. It was established in 2014. In 2017, we released the first version of AQUA-DUCT software, and the recent version was released just a year ago. And because of the increased functionality, we said that it would be a good idea maybe to introduce to you more what it can be done with AQUA-DUCT. So this is our team, and there are also contact information to us. The website information, our partners. We hope that you will be soon in this group as well. There is also information about funding agencies which support our research. And this man below the group picture is the main developer who was responsible for the coding of the first AQUA-DUCT session, and it's like a father of code of the AQUA-DUCT. And before we will start with the scientific part, let me invite you to Rinoji garden, to Zen garden in Kyoto, in Rinoji temple. And this is very famous garden. Probably most of you heard about them. It's only has 5 group of stones, and a lot of people is coming there and relaxing at the front of this open space. And there was a several theory why it happened, what it has such impact into person. And one of them was described in Nature in 2002, and it's just saying that the brain is filling the empty space between the stones, and this space is filled by the shape of the tree. So in our imagination, we are moving into the forest and we are feeling relaxed. So now you probably ask, what is the common between the Zen garden and the protein? So the answer in here is that the empty spaces. So in fact, all macromolecules and proteins have a lot of different intermolecular voids, cavities, pockets, tunnels, channels, however you call them. There is a complex network or all of these features, and they are, of course, dynamic features. They are all the time reshaping, changing the volume and the surface. They are also surrounded by different amino acids with different chemical properties. And they are extremely important for protein stability, for the ligand transportation, like the tunnels or channels. They also correspond to ligands recognition, to enzyme catalysis, and also if you want to inhibit some enzymes, most probably you will target some cavities or voids. more than 64% of enzymes are have tunnels. So majority of enzyme is equipped if in these large volumes, and of course cavities and voids are all has. So the problem which we found at the beginning of our study was that there was a lack of the universal tool which is able to consider in the same time all these features. So the geometry, the dynamics of this geometry, and also the chemistry and physics of these cavities, and to give us the full picture of this whole empty or the interior of the proteins. So that's why we are searching for how to solve this problem, and we decide to use the natural MD simulation and the natural environment, which is where the protein is located during the course of the MD simulation. And so each solvent molecule it can be used as a molecular probe, and since it's able to penetrate the protein core, so it's sampling the interior and provide us the information. So the only way to get the information was how to track these molecules and how to get the picture. So we designed AQUA-DUCT, and AQUA-DUCT is a tool for exactly for the analysis of macromolecules during the course of molecular dynamic simulation with deep insight into the protein's functionality. And we are reversing a little bit the way how to analyze the proteins. So this so we are, instead of describing the atoms, protein atoms movement, we are describing how the solvent molecules are sampling the space, the interior of the protein. And from this picture, we are trying to get information about the functional amino acids inside proteins, about the flow and direction of the transportation which occurs inside the protein. And this provides us a lot of information which you can use to apply for different purposes. So for protein engineering, which comes quite natural, that of course you can, having this information about different voids and the tunnels, you can search for for example, the gating residues, or the thing how to reshape the cavities, how to merge some cavities, or how to open the new pathways. You can also use this information for the tracking for personalized drug design. This is possible because the single amino acids substitution will provide completely different picture of the local distribution of the density of solvent inside protein, and you can track some rare events and use this knowledge for example, engineering the enzyme accessibility of the active site. And you can also use the AQUA-DUCT to for modification of the selectivity and activity of the enzyme. And here I would like to show you just two small examples of use of the AQUA-DUCT for drug design. So the first one is from human epoxide hydrolase, where we did study of mixed solvent dynamics. And what you can see on this picture is that the hotspots which were detected by AQUA-DUCT, they are most of them, or some of them, are overlap with the inhibitors which were known for this enzyme, but some of them were in surroundings, so they can provide the information about the other volumes which can be filled by functional group of new drugs. I will describe this a little bit later in more details. And the second is very recent study of MPRO protein from SARS-CoV-2 virus. This was one of the very first computational study in this subject, and we use AQUA-DUCT for two purposes. The first one was to investigate the active site plasticity. So we found that this active site is it's very flexible, and it can reshape, change the shape very much during the even short MD simulation, and that's of course the quite bad news for docking and drug searching, because the conformational space is quite high, and it's difficult to pick the proper one for docking. And the second purpose of use the AQUA-DUCT was to search for alternative binding site, and we found several different binding sites which are on the surface of M-Pro protein, and the most important was one which is also functional, and it's responsible for dimerization of this enzyme, and the monomer is inactive, the dimer is active. So if we prevent the dimerization, then of course we can probably also stop activity of this protein. So what we would like to transfer to you or introduce you during this 2 hours of webinar is at the beginning I will show you a few more some like appetizer, the real results of the analysis. Then Carolina will provide you with introduction to AQUA-DUCT software requirements and some technical part, and also with basic analysis of the solvent tracking. And then Maria will provide you with the tutorial on analysis of local density approach, and merge this information with tracking solvents, so more advanced analysis. And beside that, we will we have all the time open chat, so you can ask a question in the chat, and to our colleagues Piotr and Agata will answer your question during the webinar. The whole presentation and the record of this webinar will be available on our website together with all questions and answers. So if there would be no enough time to answer to all your questions, then you will find the answers on the website. So now let me show you the first example. So what you can see here is the cross section of epoxide hydrolase, and you can see that there is a kind of tunnel which is crossing this protein. In the middle, you can see the orange wireframe, which is representing pointing the active site of this enzyme. So, of course, from this picture, it's quite you could see quite simple that there are 2 entries which can provide the access to the active site. But of course, the question is if there are only these 2, and if you would rotate this protein, you can see that this active site is visible by 2 holes at least. Maybe here the third one, so you can ask question how many entries is related to this protein. So for that, we are using the basic analysis which AQUA-DUCT allows. So we are tracking the water molecules, and we can get the picture of the location where the water is entering the protein surface to reach the active site. And you can see from this different colors that we are able to identify 4 entries to protein by which the water molecules can reach the active site. And Carolina will provide you with information how to get this, how to receive these results, and more how to analyze the particular paths, how to see the statistical data, what is the number of water coming through particular tunnel exit, and also which of them is more important and more occupied. So that's the first results, and you will get the step-by-step introduction how to use AQUA-DUCT to get this result. And the second example, which will be shown by Maria, will be about the will be used for more in drug design, which can be used more in drug design. And this is again the picture of epoxide, of human epoxide hydrolase. I already showed these results on slide. Here you can see the protein in cartoon. In surface representation, you can see the interior cavities, which are in crystal structure, and you can see by stick representation the number of inhibitors, of known inhibitors, which are occupied the protein interior. And the problem with these known inhibitors is that most of them are with low solubility in water. So so far there is no known drug for this enzyme. And our goal was to search for new cavities which can host polar molecules, and by this way could be used as an anchor for targeting additional functional groups, and by this way to modify known inhibitors and to provide inhibitors with higher selectivity. And you can see that from this the hotspot analysis, which provides us the access to information about the location in protein where the local density of particular molecules were the highest, give us several, which of course overlap with known inhibitors, but also can provide us few, like this one from acetonitrile, and for example, this one here from water, and other from acetonitrile, which are out of known inhibitors and can be used as some indicator of location when we can try to merge new functional group and combine with the known inhibitors, and by this way redesign inhibitors and find a new one which with increased solubility and still able to bind with target protein. So I think from my side this is the all, and I will give voice now to Karina, and she will introduce with the basic tutorial and basic feature of AQUA-DUCT. Thank you for now.
Thank you very much, Artur. Hello, everyone. My name is Karolina, and together with Maria, we will be your guides in the world of AQUA-DUCT. And since my boss is sitting next to me and listening, I'm going to do a short technical introduction about what is AQUA-DUCT. And I promise I will keep it short. So AQUA-DUCT is a fully Python 3 environment software, which requires following libraries, but have no fear, it installs automatically. All you need to also have is the PyMol software, which is fully open sourced software. So you need it to generate visualization of your data. And AQUA-DUCT is running currently under Linux, Windows, macOS, and OpenBSD systems. And AQUA-DUCT comprises of 4 different components. Well, the first one is the graphical user interface called Valve Configurator, which enables the user to create their own config file for further analysis. So then we have a valve module, which enables the basic solvent flow analysis, which I will talk about later. And then we have Kraken, and Kraken allows the user to provide the statistical visualization of the statistical data. And then we have Pond, which is the a little bit more advanced module. And this, well, Pond provides the user the ability to calculate hotspots and energy profiles. And then, as I said before, we have PyMol to generate the visualization of the results. So what the user actually needs to provide, it's the trajectory file and the topology file. And you need to keep in mind that you need to prepare your trajectories in a special manner. So you need to keep the information on both the macromolecule and the trace molecules inside your trajectory and topology files. You need to, the trajectory should have fixed periodic boundaries conditions, and the macromolecule should be centered in the solvent box. And you need to also reduce the rotational movements of the protein and align all structures to a reference. So then you will have a clear picture of how the solvent flow occurs. And you also need to remember about saving the coordinates of the trajectory sufficiently frequent. So we recommend here, we recommend the ratio of saving each 1 picosecond per every 100 nanosecond simulation. And also, please keep in mind the protein hydration before even running MD simulations to ensure the proper equilibration of the system and to be able to track solvent molecules during AQUA-DUCT analysis. So we have prepared the dictionary here to explain the difficult terms we use during AQUA-DUCT analysis. And this is a really beautiful scheme, but I prepared the PyMol sessions here to better explain this for you. And of course, the presentation will be shared on our AQUA-DUCT webpage. So here we have the protein of interest. And as you probably remember from Artur's short video, during molecular dynamic simulations, the solvent can penetrate the macromolecule's interior multiple times. So we need, the AQUA-DUCT needs to limit the calculations to two very important regions. So the first one, it's the object. So AQUA-DUCT traces every molecule, solvent molecule, which enters the object. So that's the specific cavity here. In our case, this is the active site. And the tracking is limited only to a certain certain area. We call this the scope. So usually the scope is the whole macromolecule. Then, as you probably remembered, Artur showed you some colored balls and called them clusters. So, well, technically each ball is the inlet, and inlets mark those events when solvent molecule, the traced molecule, enters the macromolecule or leaves it. And then this is marked by an inlet. After the end of tracing by AQUA-DUCT, all inlets are then gathered into clusters. And this is something really important to make, because clustering is mostly an art for me. And you need to remember to keep the clusters in that way, so they will reflect the entrances or exits to the cavity of interest here. We have also something that is called cluster area. It's those rainbow shapes here. And the reddish color marks the most dense region of all inlets in the cluster. So this is also what AQUA-DUCT can do for you. And also, as I said, AQUA-DUCT traces every solvent molecule that enters the macromolecule. And we are able to show you the exact path that solvent molecule made during the course of molecular dynamic simulations. And as you probably see, the paths are color coded. So the red lines represent the entrance to the active site, to the object. So yeah, and then we have the blue parts, which represent the movements of the solvent molecule when they left the object. The object is here, so they leave the object out using the one of the tunnels to leave the macromolecule. And then we have the object part of the path. So the green lines represent the movement of the solvent molecules when they are in the object cavity. And yellow lines represent the moments when the solvent molecule leaves the object, then wanders around the protein and then goes back to the object. So they represent the available volumes for small molecules to penetrate, and the incoming and outgoing parts of the paths, they represent the shapes of particular tunnel. So we also, along with the raw paths here, we have smooth paths. So AQUA-DUCT is have some smoothing algorithms implemented, so the user also can have the smoothed path here. And okay, so this is this. So now we're going to do, well, this was the valve related part of the dictionary, and the pond related part of dictionary is here. So pond is using the combination of the local solvent distribution approach merged with the solvent transportation analysis. And it basically works like that. So, so pond divides the protein of interest into small cells, grid-like, and for each cell it calculates the density of those traced molecules. And this is how, yeah, this is something I need to show you, because then everybody will understand that. So this is how we get pockets, and pockets represent the regions of the protein which are available for small molecules to penetrate. And we have two types of pockets. So there's the inner pocket. So this represents the more dense regions of the macromolecule. And we have the outer pocket, and the outer pocket represents the maximum available space here for solvent molecules to penetrate the protein here. And also when we combine the local solvent distribution approach with the solvent transportation analysis, we can have an energy profile. An energy profile shows you the free energy values along selected path. So you are able to detect some energy barriers here. So this is it. Yes, I forgot about one thing. And yeah. So along with the pockets and energy profiles, we also have hotspots. And Maria is going to talk to you a little bit more about hotspots. I'm going only to tell you that hotspots are the most dense regions of the protein of interest for particular solvent molecules. So this is what you need to know now. So we have prepared 3 sets of data. First of all, is probably known for some people because it's a 10 nanoseconds MD simulation of mouse soluble epoxide hydrolase that is, which is available on our web page. And we have a user guide for that. And we have also prepared 2 other sets of data with human soluble epoxide hydrolase. It's also 10 nanoseconds MD simulation. And Well, the first set here contains only the enzyme immersed in water molecules, while the third set of data comprises of the enzyme structure immersed in a mixture of water molecules and acetonitrile molecules. So now it's time to do some practical stuff here. And I'm going to show you what you can obtain using the basic solvent transportation analysis. So as I said before, this is the protein of interest. Those little balls, the inlets, they mark 4 entrances to the active site, and you can check it using the surface representation, because as you can see here, there are 4 holes, as Arthur called them. So there are 4 holes leading to the active site. And if we hide the clusters here, so you can see that each hole is filled with red and blue parts of puffs, which basically means that this is used by the solvent molecules as entrances or exits. Sorry, everyone. And I'm also going to show you something more, because we can also obtain the graphical representation of the statistical data here. And with respect of incoming and outgoing paths, we can also see which particular cluster of inlets is the most used during the course of molecular dynamic simulations. And this cute plot here shows you each identified pathway, each identified events of solvent entrance or exit, sorted the way they occur during the course of molecular dynamic simulations. And we have also this pie chart representing the cluster size. And I'm sorry, I'm losing my voice right now. And here we have the intramolecular flows chart, where you can observe the flow of solvent molecules and how they use the identified clusters. Then we have scope and volume, scope, area, and volume plots. And we also have the object area and object volume plots. Sorry. Maybe this will help, I hope. Yeah, and to do so, we are going to use the, we are going to use first the terminal here to, no, it's not working. Sorry. So I'm going to use the valve configurator here. So I'm going to simply type valve config _run and hit enter. And this is the valve configurator window. Oh my God, sorry. And I'm a little bit lazy person here, so I prepared my own config file. I'm going to show you how you can use it to create your own config file. And as you can see here, we have 3 levels of editing the configuration file. It's easy, normal, and expert levels. And here, I hope my voice will let us to do that. We are going to use the easy and normal levels. The expert level here is something even I am afraid of, so I won't be using it. And Maria is going to show you the 4th level of editing the configuration file. And basically, she won't even be using the graphical user interface here. So we are going to use the easy level here, go forward. And come on, open here. Yeah, so as you can see here, we have 3 tabs. And in the initial tab, we need to provide the name of the topology and trajectory file and the name of the cache file. The cache file is the directory where AQUA-DUCT stores every information that is necessary to run the particular calculations. So we need to set the name of that directory. We usually use the name of cache. And probably you are seeing also that yellowish box here with text. And this is the graphical user interface helper. And this gives you the information how to use particular options. And so now we are going to set the scope and object definitions here. And we are going to, you probably remember, but I'm going to remind you how to do that, because we need to set the scope as the protein and the object as the active site here. So we need to type simply protein here at the scope. And the definition of the object is a little bit more complicated because we need to provide the type of the traced molecules and the information about the location of particular cavity. So now, so here we have a 6 angstrom radius sphere, which geometry center is marked by the center of geometry of residues from the active site here. And we need to also set the AutoBarber option to protein. And this is something that informs AQUA-DUCT that it needs to trim the paths the way they will fit to the protein surface. And we have a very good tutorial on path trimming on our AQUA-DUCT webpage. So we are encouraged to go there and learn something more about path trimming. Then we have the clustering tab, and we are going to use the default clustering method here. So that's the barber, and we need to once again tell the barber that we need to trim the path to fit to the protein surface. We are not going to add anything more here. And also in the last tab here, in the visualize tab, we need to tick everything that is possible. Set show molecule to protein, and then set show scope convex hull and show object convex hull to respectively protein. And in case of the object, we need to replace, we need to repeat the definition of the object, but we need to also replace the type and the name of the molecule's type with an asterisk. And I also need to tell you that those orange and purple shapes that you saw recently, they are approximated convex hull. And this is for AQUA-DUCT to just run the calculations really smooth here. So we need to save the config file. And I'm also going to save here as my config.txt file. I'm going to click save, okay, because I already saved that. And now we are going to run AQUA-DUCT calculations. So I'm going to type valve run using the flag -c. I will provide the name of the config file. So that's config.txt. And also I'm going to provide the name of the file that contains all logs, which are important mostly for the developers, but it's also easier for the user to debug all the errors that may occur during the calculations. So I need to debug file here. It's aq.log here. And I'm going to hit enter and just kill the calculations here. And I'm going to show you what you can obtain from that particular run. And you can see that we have 4 dump files here, and each dump file corresponds to each AQUA-DUCT calculations stage. So we have traceable residues, raw paths, separate paths, and inlets clustering data. So in the first stage, AQUA-DUCT just creates a list of all traceable residues. Then the list is being pre-processed using to provide the information about the raw paths, and then this is each path is written as a separate path. Then we have the information about the results of inlets clustering here, and we have the 5 analysis results files, which contains the most abundant statistical data here. So we have information, for example, about the cluster size, their areas, the probability of transfers, mean lengths of transfers, mean frame numbers of transfers, and so on, so on. So this is really, really big file. And also we have 6 visualize results script, Python script here, and the archive here. So this is those are the files. Sorry, once again. Those are the files that you need to provide to run visualization. As I told you before, you need to use PyMol to do that. So since we have the config file saved and I showed you how to run AQUA-DUCT calculations here, I will show you what is the first result we have obtained using only the default clustering method. And yes, well, this is not what we've been expected. So we have only this one big cluster. It's not separated into 4 different clusters. So we need to run the calculations once again and try to divide this one big cluster into 4 pieces. And well, the default methods didn't work because the entrances to those tunnels, to those pathways leading to the active site, are not separated enough. So the basic method here didn't work. So what we need to do now is to modify our previously generated config file. And we are going to use the normal level of editing the config file. So we are going to hit set here normal, click forward, and come on, don't be shy. Yes, so now you can see there are 5 tabs right now, and we are going to go, oh, okay, so yeah, that wasn't that config file I wanted to show you, sorry, because I wanted to show you the previous config file, so that was the config here, so we are going to use the normal here, okay, and yes, this is his second webinar, so he's probably tired right now. So yeah, this is the basic clustering section. So we are going to add something more. We are going to use clustering 2 section, where we use the Birch method to divide that big cluster into 4 pieces. And we need to tell AQUA-DUCT that after the clustering part here, where the barber tries to divide the inlets into clusters here, we need to have the clustering 2 procedure. And we need to also set the recursive threshold. And the recursive threshold needs the information about the cluster size, the size of the cluster that we need to divide, that we want to divide here. So this is simply the result of division of 1118 by 1118 121. So I assume that if I type more than 0.9, it will be perfectly fine here. And what you need to also remember that in the inlets clustering tab, there is a max level option here. The GUI takes care of it because when you add some clustering sections here, you need to also increase the max level. This is the information about how many clustering procedures needs to be run to provide the perfect results here. And I need to also change the execute here option to set it to run, because when you have longer calculations and longer simulations, you don't need to repeat all calculations when you only need to change the clustering procedure. So by setting execute to run, you tell AQUA-DUCT that you want to start your calculations from the clustering section here, clustering stage. And those 3 stages, well, AQUA-DUCT needs to only load it and keep them in mind. So we need to save the config file here as config2.txt. Yes, I know that. Thank you. And yes, and we are going to, well, we are not going to run that. Oh, and also one thing before I forget about that. Because we want to override, because we need to run the calculations once again, we are going to override the files from the 4th stage, and the 5th and the 6th as well. So, well, we recommend you to keep those files stored in some safe place. So I have like several directories for that. And so after the 2nd run, I can show you that the results of the division of that one big cluster into 4 pieces, well, that wasn't something we were expecting. So when you show the protein as surface and add some transparency to that, you can see that, well, there's need to be some adjustment done. So first of all, we're going to split the cluster 5 into 2 pieces. So then we have that side cluster reflecting the side entrance here, and this gorge part of the cluster. And then we are going to, we are going to split the cluster number 8 into halves. One half would be incorporated to the cluster 7, and the other half would reflect the gorge part of the entrance here. So we are going to do that, but first of all, we need to check the sizes of those 2 clusters. So that was cluster number 5 and cluster number 8. So cluster number 5 is 222 inlets, and cluster number 8 has 226 inlets. So we need to do the math yourself. And yeah. So once again, I'm going to run valve configurator here, load the previously generated config file, the config2.txt, open it in normal level of config editing, hit forward, and open this window here. Yes. Thank you very much. So I'm going to add another clustering section here. I'm going to call that clustering 3 and use the purge method here to divide those clusters into 2 pieces. So we needed to divide 226 by 1121. So that's probably something smaller than 0.25. That should work. And we need to also tell AQUA-DUCT that we are adding the clustering 3 procedure here. So after clustering 2, where we divide that big cluster into 4 pieces, we need to run the clustering 3 procedure here as well. So as you can see, max level have increased here. So I'm going to once again save that config file, save it as config3.txt, and yes, I want to overwrite it. Now close it, and since I've calculated this previously, I'm going to show you the results here. And yeah, come on. So as you can see, it's a little bit messy, but well, after doing some small adjustments, it will be good as new. And so we have cluster 6 that's perfectly overlap with the tunnel entrance. We have cluster number 10, which also reflects the entrance to the active site. And we have cluster number 7, which we want to join with cluster number 12. And we have cluster number 9, which we want to join with cluster number 11. And we also have this 3 little clusters here. 2 of them are here, and 1 is here. They have only 1 inlet in size, and we call them rare events, so something that occurs really rare. And for the picture clarity here, we want to create something that we call cluster of outliers. So so we are going to move those 3 clusters into 1 cluster of outliers. And so I'm going to close that now, and we are going to use valve config run for the last time, hopefully. And I'm going to load the previously generated config3.txt using normal level here. And I need to hit forward. And oh, don't be shy here. Come on. Yes. So now in the inlets clustering, we need to do 2 things. Well, first of all, we are going to join the clusters. So I'm going to type 7 plus 12, white space 9 plus 11. So I'm going to join clusters. I'm going to also renumber those clusters so they will have numbers according to their size. I'm going to change the detect outliers option here to auto. So we will have outliers clusters, and I will set singletons outliers to 1, because this is by setting the maximum size of the cluster that should be incorporated into in the cluster of outliers. So once again, I'm going to save the config file here as config4.txt. I'm going to save that, yes. And well, finally, I can show you that we have those results prepared here. And when you type Python in the name of the visualization script, then you are able to visualize all available items here. So well, the generation of this final session is quite fast. Well, it should be quite fast. So I'm going to wait for it a little bit. Yes. Okay, yes, so this is it. This is almost everything. So we now have our protein of interest and 4 clusters reflecting the entrances to the active site. And what is the most powerful thing that AQUA-DUCT have is that it is able to present the pathways of all small molecules that that entered the scope here and visit the object. In general overview, so we can show the pathways of molecules that entered and left the active site using the same tunnel, or using two different tunnels. And also we are able to tell which way they entered and which way they left. So we are able also to show it the other way around. Also, as I said before, during the dictionary part, we can show the smooth paths here. So also using this cluster as the entrance and the exit. The same situation, so using this cluster as the entrance and this as the exit, and the other way around. So this is it. And also, what I forgot to mention, we have the ability, or AQUA-DUCT has the ability to show you the master paths. So master paths represent the general overview of how the small molecules were moving when they entered using this this entrance, and when they left using this exit. And also, as I showed you before, we have the we can gather the incoming parts of the paths, outgoing parts of the paths, the object parts of the paths. So they so yeah, you can also visualize this, and we can see every path here as a separate primal state here. So you're able to visualize all available, all identified pathways, and also as well as raw, but also as smooth pathways. So this is almost it. I'm going to also show you something. First of all, I'm going to close this because it's getting a little bit tired of all these primal sessions. So the last thing I want to show you is the Kraken. So I'm going to type Kraken_run. In Kraken, as I said before, this is the graphical user interface tool also, so where we need to provide the name of the output file, so that's results2.html, and load the 5 analysis results.txt file. So we are able to visualize the information about the cluster size, molecular entry time distribution, intramolecular flows, clusters area. And when we load the CSV file also, we are able to see the relative cluster flows and volume per frame flow plot. So I'm going to run this now. Yeah. And I'm not going to wait for that, but I'm going to show you that this is the resulting file here. So you have the cluster size information here with respect of the incoming and outgoing parts, the relative cluster flows, as I said before, the molecular entry time distribution, cluster size pie chart, and the intramolecular flows chart here. And this one represents the flow of the identified solvent molecules. And as you can see, most of the traffic occurs using the same cluster, the same tunnel as the entrance and the exit, but also there are passages using this tunnel as the entrance and the tunnel number 3 as the exit here. Also, there are movements to the cluster marked as N. And so AQUA-DUCT, well, N is not technically a cluster. AQUA-DUCT marks all pathways that have ended or started in the protein structure as N. And you need to remember that we showed you here only 10 nanoseconds simulation. So you need to keep in mind that it's relatively short. And if you will be running longer simulations, the size of the N cluster will definitely decrease. So yeah, my voice is now really killing me. So I'm going to end with this. So this is everything I wanted to show you and tell you and show you about basic solvent transportation analysis. Thank you very much. Hope you enjoyed.
Thank you very much, Carolina. Thank you for going through your part. What I would like to add here only is that this example, this tutorial could be rather difficult to follow. Of course, you can download and you can look it one more time. But we took this example for purpose. We wanted to show you that the AQUA-DUCT is very flexible and you can really use the clustering, several clustering methods, and you can join, merge, and split, whatever you want to do with clusters. And in some cases, it has to be done. But in some others, and you will see it in next example, it doesn't need any user interaction. So this is really system dependent. Also, I would like to say you that this flexibility also gives you a possibility to set your object wherever you want. So it can be active site, but of course it can be also other location in protein you are interested. So it can be cavity where your cofactor is bind, it's located, or in case of proteins with two active sites, then you can set one object in one active site, the second time in the second one. So you can play with these all things, and for that you can use, of course, one MD simulation. Only what you need to do is to run AQUA-DUCT more than one time and to analyze properly the results. So it gives you, of course, the information about features which are not accessible by other software so far. And now I will give the voice to Maria, and she will let you know how to use the pond and the other analysis. Okay, so Maria.
So hello everyone. My name is Maria, and yeah, as Arthur and Carolina mentioned, I will guide you through the rest of our sample data. So the first set of the sample data is the 10 nanoseconds MD simulation of the human soluble epoxide hydrolase immersed in pure water molecules solvent box, while the second set of our data is also human soluble epoxide hydrolase, but this time it will be immersed in a mixture of the water with the acetonitrile molecules. So let me just close all these windows and I will go to the proper directory. And yeah, first I would like to show you, as Carolina started her tutorial, let's say, the final result that we can get from the combination of the valve analysis as well as the pond analysis. So we just need to wait a while to open this session. And yes, here is the final results from AQUA-DUCT. And as you can see, what we have here, it is kind of similar to those what we had previously. So we have our protein of interest. We have 3 clusters, which indicates the entrances and exit areas to our active site in the protein. But what we have here more is the information about the hotspots, which are marked as spheres, and also information about inner pockets, inner and outer pockets. So the actual volumes which can be occupied by water molecules. And here I also want to show you that we can obtain the information about two types of the hotspots as well as the pockets in our protein. We called these two types of hotspots. The first type is the local hotspot, while the second type is global hotspots. And what is the difference between these two types of the hotspots? So first of all, the local hotspots, they are calculated only for those trace molecules which entered the scope, but also entered the object area. So in our case, it is the active site. And the second type of the hotspots we can calculate, they are called global hotspots. And as you can see, they are placed, they are located within the whole scope of our protein. So here we are also taking into account such pathways during the analysis, which not necessarily entered to the our object of interest. So AQUA-DUCT is also able to take into account all the other pathways which went through the scope of our interest. And what is also very nice, and what AQUA-DUCT is also able to do, is to provide such an analysis in time window. So what is a time window? We can divide our simulation into pieces, and we can observe how the hotspots as well as the pockets change within the time. And here I divided my short simulation into 5 windows. And as you can see, we have 5 different states. And yeah, we can say a little bit more about the evolution of such features within the time. And as you can see, the places of the hotspots, they can be placed in different places in our protein, and they can change. Yeah, here you can also observe that our hotspots, they are all spheres, but they have different sizes. So here the size reflects the local density. So in places where the local density is higher, the spheres are marked as bigger. And what also POND is able to do is, as Carolina mentioned you during the technical part, let's say, is the ability of calculating the energy pathway of the particular trajectory of the molecule of interest. And here I will also show you how to obtain such results. So this is our final session. And yeah, as you probably remember, because POND calculation is actually merged with the basic solvent transportation analysis, first thing we need to do is to run the valve calculation. And Carolina already showed you how to prepare your config file using graphical user interface. But I will show you something else. I would like to show you that it is not necessary to use graphical user interface. You can use just simple plain text file, simple config file. And this valve from AQUA-DUCT is smart enough to create the template config file, and how we can obtain such template that we can easily modify. We just need to type valve_run, and we need to use the option dump template config, press enter, and as you can see, we have all information that we need to provide in the config file. And these options, they are as in the expert mode, expert level of the configuration in our graphical user interface. And I will use this as a plain text file. So first I would like to save this template. So I can call it config_template txt, and it is saved in this directory. I'm here, so you can see this is this config template file. I can open it, yeah, and you can see that we have all the information, and I already prepared the proper config file, and right now I want to show you which options I had to change to provide good config file. So I will use the smart program to compare my template file with the actual config file. Oh, and yeah, you can see which options had to be changed. So first of all, we need to provide, of course, the topology and the trajectory. You probably remember that from our presentation. And because I will launch the calculation in the directory in which I stored my topology and trajectory file, I just need to type the name of those two files. But just remember, if you are launching calculation from some other directory, you just need to add the absolute path to those two files. As Carolina mentioned, the cache directory is also very important. So in this option, cache_dir, I will type cache. So the valve, the AQUA-DUCT, will create the directory with the name cache, and AQUA-DUCT will store all the necessary data. Here again, we need to set our scope into protein, and because we are using convex hull approximation, we need to set this option to true. And yeah, as Carolina mentioned previously, we need also to provide the definition of our object. So here, once again, it is the active site in our protein. So here we have the residues from the active site. We are tracing only water molecules. So the selection is res name W18. And yeah, I set the center of the geometry with the radius of 5 angstroms within these residues from the active site. And here it's something new. This is option add passing. And this is this option which is crucial for pond calculation, for global view of our hotspots as well as pockets. So we want to track, again, water molecules which visited this code, but not necessarily the object. So we need to set this option add passing into res name W18. In the second stage row paths, I won't change anything. And in the third stage, I just set the auto-barber option into protein, as Carolina explained you in the previous tutorial. Yeah, and as you probably remember, in the previous case, we had this picture of some clusters which size was only one inlet, and at the very beginning I want to avoid such a situation, so I just set the option detect outliers into true already, and the singleton outliers to 1, so we will get in the first run of the clustering the cluster of outliers. And here in the visualize section we will change some options to true, because we want to visualize our raw paths, smooth paths. We want to have also the states of our paths, so we will be able to observe our trajectories of the water molecules one by one. I also want to see our macromolecules, so I will set this option to protein. I also want to see my convex hull of the scope, and I also want to see my object. So here again I had to copy the definition of the object, and I had to remember about the change the name of the trace molecules, and I had to change this into asterisks. And actually that's all. All other options are default options, as in this template config file. And right now, yeah, we can close this, and we can just simply run our calculation by typing valve_run with this flag -c and provide the config file, and again the debug file aq.log. Yeah, and we can run our calculation and here, yeah, I will also kill the calculation because it will take a while. So I already calculated all the necessary data, and as you can see, we have once again the dump files, as well the 5 analysis results, and the visualization, the script for the visualization. So yeah, let's see our results. Yeah, I need to type here Python 3. And I just want to show you the clusters, molecule, object, and let's say scope. And we will see if this default clustering was good enough. And yeah, as you can see, what we have here, we have our molecule of interest, yeah, scope, object, they are perfectly fine. We have our outliers clustered together, and the 3 clusters which mark the entrances and exit area to our protein. And you can remember all these things from the previous tutorial. And right now we will go to the pond related calculation. And for pond, we don't have any graphical user interface. We will just use our terminal and command lines, and we will write commands. So first of all, how to run pond calculation. So it's really simple because we just need to type pond_run. And you should remember that, of course, we have some little helpers. So once you write help with the pond run, you will get the information about the possible options that you can use and you should use to obtain the desired results. And what we want to obtain right now. So first of all, we want to obtain the information about the pockets and the hotspots. and what we need to provide with this command line. So let's start with the config file, because Pond needs to read our previously prepared config file. We also want to provide, to create actually some log file in which we will have all the information about the calculations proceeded with the Pond. And we also want to create the new directory in which we will store our calculated results. So first of all, I want to calculate the pockets and hotspots only for those molecules which entered the object area. So this is why I called this directory local. And yeah, we want to calculate pockets and hotspots as well. And we also need to calculate something which we called reference value. And for calculating the reference value, we need to use the option reference calc. And because why we want to calculate any reference value? This is for the resizing purpose. And if we want to reflect the density of the particular hotspots inside our protein, we need to calculate something which we call reference value. And the reference value, it is calculated outside the scope, so in our solvent box. So in our solvent box, we have only water molecules only. So I will use the option reference mol and type res name WAT because it stands from water molecules. And the type of our paths is also water molecules. So I will use the option paths types and type here WAT. Yeah, and actually we can also use the option output suffix, and by using this option, Pond will add to each of the output file the suffix local, because it will help the user indicate which files are which, in case you are saving all your output files in one directory. And right now we can calculate our hotspots and pockets, and it will take maybe 1 or 2 minutes. And I want to show you what the results will look like. So let's wait this 1 minute. And as you can see, we have the reference point in which the Pond will calculate our reference value. So actually right now, Pond is calculating the reference density in this reference area. So 30 seconds more. And right now it's, let's say, very fast to calculate. And of course, when you will proceed with longer trajectory, it will take a little more time. But here it's almost done. And yeah, as you can see, we have the information about the reference number of molecules, the reference density, and also our reference value, which is the most important for us. And here I would like to mention that if you are calculating not only local hotspots, but then you are calculating global hotspots, and if you want to proceed your analysis also in time windows, you can always use the previously calculated reference value So you don't need to repeat the calculation of the reference value every single time. And in that case, you can change the reference calc option during your calculation into reference value and provide the calculated value. So as you can see, our calculations have finished. So I will once again open the PyMol session with our clusters. And right now, I would like to open our results. So it's in local directory. And I want to open inner, outer, and hotspots in mol2 extension. And yeah, as you can see, we have not so nice picture, but in a minute it will be nicer. So I would recommend you to change the representation of the inner and outer pockets into mesh representation. Yeah, but we can show as mesh to have only those representation. Yeah, and keep the hotspots as spheres. We can provide them some color, for example, blue. And right now, I would like to resize my hotspots to reflect their density. And we can do it quite easily because AQUA-DUCT provides a script created for that purpose. And we can easily find this script. This script is called hs_resize, and it's Python script. And here we have the location of our script. So I will copy the location. And I will use right now the PyMol terminal to run this script. So the procedure is as with any other script which is used in PyMol. So I just need to type run and the path to the script. And right now we can use the script. So as you can see, during the pond calculation, we obtained something which is called JSON file. So actually I need to provide the path to this file because in that file Python has the information about the reference value. So yeah, I provided the absolute path to this script. And I also need to provide the name of the object I want to resize. So in my case, it's hotspots_full_wat_local. And right now you can see that, yeah, the size of the hotspots change according to the density of the particular hotspots. And as you can see, the most dense hotspots are located close to the object. And yeah, you can do exactly the same thing for your global hotspots to calculate your global hotspots. So let me just copy the previously written command. And what we need to add to calculate the global hotspots? We just need to add the option row because this, sorry, I need to share the screen. Once again. Okay, sorry. I hope you can see it right now. So we just simply type exactly the same command but we need to add the option row to include those pathways of the trace molecules which enter to this code but weren't within the object area. And I will just change the output suffix into global, and as I told you, it is not necessary to calculate the reference value once again. I will use this value which was calculated previously, and I will also change the location of the saved files. And yeah, right now I can run the calculation. And as you will see, it will be a little bit faster because we won't calculate the reference value. We will simply calculate only pockets and hotspots. But let me kill this because I want to show you something more. As I showed you during this first session, we can also calculate those hotspots and those pockets in time window. So here again, it is really simple to proceed with this. You just need to add another two options. The first option is the option windows, and I want to divide my calculations, my MD simulation into 5 windows, and each window will contain 400 frames. Why 400? Because my 10 nanosecond simulation, the coordinates were saved every 5 picoseconds. So in total, we have 2,000 frames. So I just simply need to divide, I need to divide 2,000 frames into 5 windows. So the window size is 400. And we can run the calculation and we will obtain this beautiful picture of the hotspots in time window. And we can proceed exactly the same with the resizing of the hotspots. And last thing I want to show you in this tutorial is this energy profile. So energy profile can be calculated for the particular pathway. And let's say that we want to look for the path, the trajectory of water molecules which enter through the cluster number 3 to our protein of interest, and left the protein through the cluster number 2. So let's go to the row paths and let's try to find some path which is appropriate. And here, this path, it looks quite nice because we have the actual trajectory of water molecule which entered to the scope and to the object and left the protein. But as you can see, this path, it's a little bit complicated because it is a row path. So we need to smooth this path. And during the basic analysis which was conducted by Pond, we were using some default options for the smoothing. And as you can see, exactly the same path, which was that complicated, right now it looks better. But from my experience, I can tell you that this smoothing is not enough, let's say it like that. We have also additional tutorial only for smoothing, and it is also provided on our webpage. So I encourage you to take a look into this tutorial. So what needs to be done is to correct the smoothing options in our config file. And actually, I already did that. So I will show you the final config file because I just simply add one option to the last section, which is smooth section. I add the option recursive, and I provided the value 105, and it's quite a big value, and we'll obtain better smoothing using such a value. And because the smoothing, it is the stage which is connected with the stage separate paths, we need to repeat the calculation from the third stage. So here with the execute option, we will need to type run. And I already create the special directory for smoothing, and I copied the first and the second dump files, and I run the calculations, and I obtained the results, and I want to show you right now how the smooth path will look like, exactly the same path. As you may remember, it was path number 12. And as you can see, the path is nicely smoothed. And yeah, it is something what we can provide to calculate the energy profile. And here again, we will use the pond to calculate the energy profile. It's really easy to calculate the energy profile because we just need to add 3 options. First option is path ID, and how we can obtain the ID of this specific path. So as you remember, it was the path number 12. So we will go to the 5 analysis results txt file, and we will go to this big table with list of separate paths and properties. And we need to look for the path number 12. It's here. And as you can see, we have the specific ID of this path. I will copy this ID, and yeah, I will paste it here. We want to calculate the energy profile, so I just need to simply add the option energy profile. And because we are using smooth path, to do that, we just need to use the argument path smooth, and we can run the calculation. And I already did that because it takes a while. And as you can see, I obtained 2 additional files, the dat file and the mo2 file. And in that file, we have information about the length of the path and the energy of the particular part of the path. And this mo2 file we can load into PyMo session. And I already did that in the final session. And we can color this path according to the energy of the path. And as you can see, this red part, so the part where the energy is the highest, this part is located close to the loops of some loops of our protein of interest. And as we know, loops are highly flexible normally during the course of the molecular dynamics simulations. So it may indicate that these loops may provide some hindrances and obstacles for this particular water molecule to pass through the protein. So this is the place where the energy barrier is. And we have also the direct information what is the value of such energy barrier. And this is all when it comes to the data set number 2. And at the very end, I would like to show you the last but not least results. So those connected with the co-solvent analysis. And it will be actually really quick, because right now you can proceed with the basic transport analysis calculation as well as with the pond analysis. And this is exactly such a case that we once again want to combine the basic solvent transportation analysis as well as pond analysis. And here we have our final results. We have 2 clusters. And as you can see, we can indicate in those 2 clusters areas and particular inlets which belongs to specific type of the trace molecule. So because here we were tracing water and acetonitrile molecules as well, we can indicate in which places acetonitrile molecules or water molecules are entering our scope region, our protein of interest. And what is also very nice, that we can divide the particular paths into those parts which corresponds to particular type of our traced molecule. And we can do exactly the same thing for our hotspots. So we can calculate the hotspots for acetonitrile molecules as well as for water molecules. We can do it as it was in the previous case for local hotspots. We can do it also to get the global view, but this is more for the protein engineering purpose. And if we want to target some other parts of the protein, here in that case, we wanted to target the surrounding of the active site cavity. So we just simply calculated the local hotspots, and we can of course deal with that in time windows mode. And how we can analyze hotspots for different type of the co-solvent. So first of all, we of course need to run our valve calculation, because as I told you several times, this is combination of the basic solvent transportation analysis and local density approach. So in our config file, we just need to provide, of course, the proper topology and trajectory file. But everything else, it is exactly like it was in the case of the config file for the simulation in water solvent box. And here, because we have in our solvent box not only water molecules, but also acetonitrile molecules, we need to add the name of the acetonitrile to the list of the trace molecules. So here in the object, I will type res name WAT because of the water, or res name ACN because of the acetonitrile. And in case I would like to calculate global hotspots, because why not, we can also use this option add passing. And here once again we need to provide the information about not only water molecules but also about the acetonitrile molecules. And the rest of the config file is exactly as it was in the previous case. So we can run the calculation as it was previously. So valve _ run, provide the config file, and we can create the debug file as well. As for the pond calculations, we will just use once again the pond run command. We will provide the config file, the debug file as well. I will type pond underscore WAT, because first I would like to calculate the features for water molecules. I just want to create the directory in which I will store my results. I am calculating pockets and hotspots as it was in previous case. Of course, I need to calculate the reference value for the resizing purpose. But what is here very important, that I need to remember that I have in my system water molecules as well as acetonitrile molecules. So I need to use res name WAT and ACN for reference mole option, and for paths types, because first of all I am calculating the hotspots and pockets for water molecules. I need to type paths types WAT, and I can also use this output suffix option and type, for example, local, because we are dealing only with the local hotspots and pockets. And I just simply need to run the calculation. And right now I will kill it because I already calculated it for you. So as you can see here, I have my directory pond results, and here I calculated the local hotspots for water molecules. I did exactly the same for the acetonitrile molecules. And as you can see on our session, I already loaded these hotspots as well as pockets into session. And yeah, the location of the hotspots is different according to the type of the traced molecule. And yeah, you may ask why, because actually we have the polar water molecules, the polar acetonitrile molecules, but of course these two types of water molecules, they are different from each other. Their shape is different, so they can also penetrate some different places in our molecule of interest. So for example, here in that case, water molecules, they are placed mostly within the object area, while the acetonitrile molecules, the hotspots, the most dense areas are in the region we called, in that case, cap domain, and we have also some hotspots in a core domain. And maybe as you can remember from the session that Arthur showed you, the known inhibitors, they are placed mostly within this object active site region. And here, by using these different molecular probes, we could identify places which are different, which were not targeted previously. And as I can tell you that we can use different type of molecular probes, we can run multiple simulation with different co-solvents. We can run the hotspots analysis for all of them. Then we can merge our data, and we can probe our protein of the interest with different molecular probes, and we can get an information about different region in our macromolecule of interest, which should be or may be targeted, for example, during the drug design process. And I think that's all what I wanted to show you during these two datasets analysis. And now I'm giving the voice to our chef.
Thank you, Maria, for our presentation. I hope that this brief introduction to pond analysis will be useful. There would be now the time for questions and answer sessions. Few questions appear already, and we try to answer them. I want to maybe stress one more thing, which is quite important, that we wanted to design the AQUA-DUCT in the way to be as much flexible as it's possible. So to be able to answer several questions using this approach. So the combination of the tracking of small molecules and the local density calculation and analysis. So therefore, there is several also modes of action you can use AQUA-DUCT. So the simple one, which was shown mostly here. So this is a standard mode. So it means that you have single MD simulation and you are analyzing the single trajectory and you get results. But what already Maria showed you, you can use also time window mode. So it means that you can separate your single simulation into some steps, and they can overlap or they can be consecutive. So it depends on you. And by this way, you can analyze the evolution of your protein, and perhaps you can detect some sets of different conformations which can provide, for example, different functionality. So you can, for example, imagine that particular conformation of protein makes access to active site easier. So it's open state and closed state. Or you can, for example, imagine that the binding cavity or the active site cavity or the cavity where you are targeting for your drug design can change and make the also other shapes with a different location of the hotspots. So you can try to figure out that one. And also we add other type of mode. So we call it sandwich mode. So it's mostly used when you have several short MD simulation and you want to average them. So instead of running one by one, you can use the AQUA-DUCT sandwich mode to run analysis of multiple simulation at once, and you can get access to like overview of the statistic of all simulation simultaneously, but also you have the information from each separate, so you don't lose this data. And other last, the mode of action is we call it considerator mode. So it's for basically it's for analysis of the rare events. I mean, by rare events, we call something what occurs once or a few times during course of the simulation. So the statistically, it's not enough usually to perform some analysis. And what we can do with this mode, we can cut the particular piece of MD simulation where this event occurs and merge together, so to build like one long simulation where repeated are these rare events, and then analyzed by the same way like the standard during the standard mode. So basically, our aim when we were designing AQUA-DUCT at the beginning was just to track the water molecules, the solvent molecules, and then we were adding some functionalities. So now it's merging this simple tracking system with the analysis of the local density and with calculation of the energy. And we are working still on some other features, so to provide you more features, for example, for drug design and for the analysis of different hotspots. But so I think that's all from our side. And if there would be any questions, we are open. So you can type the questions in chat, and of course, if you will have later on questions, so please don't hesitate to write an email, and we will. Okay, there are some questions. Yeah. Yeah, the question concerning, I mean, if we discuss about the design of the calculation for the specific system, it's really important. The AQUA-DUCT really requires some knowledge of the system. So you need to address the specific question you are interested in. And so for example, if you have the complex system of the protein and membrane, you can use you can limit your system to analyze only how the solvent pass through a protein, but also you can build as a scope both the protein and membrane. So here you are not limited. So it's just depending on the question you set, and then you can design your system. You have full flexibility to put the object and the shape of the object. It doesn't have to be like we show only the radius sphere. It can be even created from a few objects.
And I have some suggestions here, because in the transmembrane oligomer here, you might like to define the scope as the oligomer, but also use the scope inflate option. So probably it would be better if you just contact us on info@aquaduct.pl on our mail, and the support will eagerly help you. So.
Yes, exactly. So I mean, if you have any specific system-specific question, it would be easier for us to answer once we will see your system. Here, I only want to underline that you really are very, I mean, the system was, the AQUA-DUCT and the software was developed to be as much flexible as it's really only possible. So we hope that we can try to answer most of the questions you will ask. Of course, not all, that's for sure, but quite a lot.
But we can find the answers together.
So we are just on time. It's 7:05 here, so we have like 2 hours perfect from the starting. So yeah, thank you very much, and it was pleasure to host you and to provide you with our webinar. And I hope to contact. Yeah, there will be a certificate of attendance. If you only need, just please write the email to us, and we will provide you with certificate of attendance. So one more time, thank you very much, and hope to see you in future, and good luck in your research. Thank you.
Thank you.
Thank you.